the urease. Because urea is a protein and it's put it into God clone. Okay? Destroys it. So for membrane filtration, we use it for two reasons. To filter, sterilize urea, to do the urease test, and also to collect coliforms. If you collected coliforms, do you remember what this is? E-N-D-O. You took the membrane, which was white at that time, put it here, put the pink membrane onto it, well, pink media, and if you have a metallic green sheen, you had coliform in color. Coliforms, if you filtered 40 mils, you could calculate your organisms per mil. You just divide by 40. Excuse me. If you did 100 mils, divide by 100. But you don't report coliforms and number of organisms per mil. How do you report it? Per 100 mils. So first calculate them to organisms per mil, so you won't confuse yourself because you may have filtered 30, you may have filtered 25, you may have filtered 75. And then multiply times 100, so it would be a number of organisms per 100 mils. That's the standard way you report coliforms. What is coliform? E. coli is what indicates presence of fecal matter. Definition of group is from the intestine, gram-negative rod, positive for lactose fermentation. Yeah, lactose fermentation. Now, talking about lactose fermentation, what are these two places? Conkeys and EMB. If the organism can ferment lactose, it will take the color into the colony itself, and this will turn red, this will turn dark red to black, and with the one exception, that metallic green is any color. If it's colorless, translucent, dirty white, or actually turns this yellow and there's no pigment, that is a what? No lactose ferment. Not a what? Not a coliform. So I'm going to project out. One, what was it used to grow? Two, what's in it that makes it special? Alkyl is a selective agent to grow what? Mycobacterium. What is mycobacterium? Tuberculosis. Okay. Slow grower. Mycolic acid. Okay. What else is it? Somebody else say it. Leprosy. Okay, it also has egg whites in here to help with the growth. To help it grow better. Slow grower. You did not have leprosy, and you did not have pathogens handed to you. What was the one in the two that you dealt with? Mycobacterium. Think about your dog and cat. Flea. It's spelled P-H-L-E-I, but it's pronounced flea. Flea. So remember that one. When you took this one, I'm going to show you a picture in a minute. This nice black one. You did an acid-fast stain. That was always for mycobacterium. You did it in the hood. You used moist water, boiling water, and you put what dye on filter paper to dye this organism. Carbofusion. So these are hot heat. If you have a skin sample handed to you, skin, and you find acid-fast organisms, also called the what technique? Seal-Nielsen. Seal-Nielsen technique. You see those hot peak organisms and you've done the staining technique. What is your disease? Lesion on the skin. Leprosy. Okay, what if it comes from in the lungs or sputum? Tuberculosis. Sometimes you want to check out your source. Okay? For tests, we're going to go to the biophysical test. We're going to go down in a minute. Okay. Don't confuse fetal red broth test with an added carbon source with the methyl red test, because they are absolutely opposite. opposite. The phenol red test starts off sort of red or red orange. You hear the Durham tube sign. That is a Durham tube. If the organ you put a sugar in this, we put in glucose and another one we put in lactose. You can do a whole range of these. You can do lactose, fructose, you can do sorbitol, you can do mannitol, you can do mannose. But you can do in a whole array. But we did glucose and we did lactose. Positive produces acids and changes the color indicator to yellow. So which one will be positive is the yellow. Also note some of the organisms. Like know what organism is positive for urease. Know what organism was positive for citrate. Know that. The Durham tube is for what?
Gas.
Gas reaction. Gas reaction. Moving to the methyl red. The methyl red test, if I have to go over to the word for the BP, starts off, it's sort of a just straw colored test tube. You add what reagent? Methyl red. They're the droppers over to the side. That's why they're there to look at. If it's positive, it will turn reddish in some form. In this case, you're looking for end products, not for what carbon source it has. This means mixed acid production. In this case, acids means red. Completely opposite from phenol red. If it's negative, it pretty much looks the same. If you're heading for VP, which is most Proxnar, and I'll have an example here, but I'll put one on the board, you add two reagents. What are the two reagents you add? KOH creatine, alpha naphthol. And in the chemical reaction, they will detect what? Butane-diol. Butane-diol. You get a red ring at the top. If it's positive, you get a red ring at the top. If it's negative, it can stay green or straw colored or no difference. I'll show some pictures of that later. Sim. Stab it to the bottom. Sulfide, indole, motility. If it turns black, there is positive sulfide production. You know what organism that does that. If you drop on it what reagent, what reagent to use for the indole part, sim? Covax. G-tax, indole. Will be a red ring again at the top. Red ring at the top. Okay. Motility, they're all local. Citrate, can it use the carbon source citrate? Very similar in a way to phenol red. It wants to use the carbon source, but in this case it's a slant. Green. No. If it can use citrate, it turns this royal wood. Okay, who are left out? Urease. Starts off sort of pinkish straw color. Remember you filter sterilized it. If an organism is urease positive, it turns hot pink. Okay, but what happened? Urea was in the solution. The organism produced what? Well, yeah, it produced ammonia by the enzyme urease. It became more alkaline in this case, not more acidic. Now these are the test tubes for us to learn. We don't have hundreds of test tubes in lab. So over there on the side, you'll see the microtiter plate, you'll see the integral tube, you'll see the API strip, and you'll see the Vitek strip, which actually combines these and many more tests with color changes, which we use in a lab to identify organisms. Of course, the goal is to identify organisms. That covers that part. This is a what plate? That is this plate. Sort of a plum color. Black ferment, not a black ferment. Now, there's something different here though. This is positive lactose fermentation with diffusible pigment. These rocks because it's spread out. This is positive lactose fermentation with no diffusible pigment, not water soluble, which means it has to be liquid soluble. This is simply what? Lactose fermentation. Which two of these could be columns? These two could be columns. Yellow, by definition, cannot be a column. Cannot be a column. And remember that metallic green sheen on the UV. Wheel feelers body reacts to corresponding antigen and gets glutination. Tiny little granular appearance, not a color change. Tiny granular appearance down the fluid in black spots. What was the organism? Proteus. What does Proteus cross-react with? Rickettsia, which calls, in this case, it's Rickettsia for what? Typhoid. Okay, how does that relate to World War II? Do you remember the doctors that saved the Polish towns by inoculating the people in the towns with Proteus? They thought they all had typhoid, they didn't take them to the concentration camps. Saved a lot of people. Okay, heading for the rheumatoid factor, glutination. But what is attached to the antigen that you saw here on the black little strips and you saw the white clumps? What makes it more visible? Latex. Is latex attached to the antibody or the antigen? Antigen makes it more visible. Be able to see those and tell me, well, real Felix reaction, glutination, Proteus, typhoid for rickettsia, latex on the antigen for rheumatoid factor. Remember those. Complement fixation, don't worry about it. We didn't have a good example at this point in time. Zone of inhibition, when you measure a zone of inhibition for an antiseptic, you measure what? From the edge of the disc to the edge of the zone. Edge of the disc to the edge of the zone. In other words, it's a partial radius. For antibiotic, which is also called the Kirby-Meyer test, how do you measure the zone of inhibition? Entire radius entire diameter are the radius times 2. This is a zone of inhibition. This is your antibiotic plate. You'll be measuring edge out, entire diameter, on the radius 2x. Two of those are correct. What would you measure? How would you measure that zone of inhibition? The entire diameter or 2x the radius? 2x the radius. Okay? This is your unit of diffusion point, where you see the pale white line. That's going to show complementary antibody to antigen with the formation of the linkages. And it's going to be pale. Put it up to the light, put it to a black background, whatever helps you see it. Okay, know your basics. Look at your columns. For instance, when we start talking about vectors today, and I'm talking about allowance or lice, it's not going to be under old version. In fact, it's going to be on the lowest power. Same thing for worm eggs, protozoans. They're not going to be under the highest old version. If I have a mycobacterium under there, it's probably going to be under old version. Remember your dimorphic fungi. Who's your dimorphic fungi? That's the pathogen. That guy. Candida albicans. That's the only one we talked about. Know what it's idioform, Aspergillus, and penicillin look like? Aspergillus has the ball, the hyphen coming down. Penicillin has the 4 fingers. Okay, we're talking about this. Slides of related diseases. So if I don't get it all covered at the lab, I'll be here tomorrow. And also, heading to the board. Also, it's still down here. Okay, my bacterial flea, we already said. Don't forget Borrelia. Borrelia can show up here. And we're going to head for these guys. I'm going to turn off briefly, and I'll put this back over to scroll through the pictures while you have your lab. Protozoans, worms. They're going to different color. I want to make sure you keep them separate. Keep them separate, keep them separate. Basic protozoans. Cover even the amoeba. Flagellates are mastodophora. I'm using the old terminology because remember they've redone everything before you to write some RNA sequences. Egg-complexes have the apex structure. What does the apex do? It's a cannon. It opens up target cells, host cells, so that they can get in. Last guy we only have one example of is the ciliate. blood cells. The red blood cells would be in him. Okay? Causes diarrhea. Malaria, this is a sad case when a person dies from in a pond. You get an entry through the nose, heads full of what? Brain. So fresh water is fatal. Anthony, again, of fresh water, like those ponds. Remember the one about the musician that went to Europe and had contact lenses, heads full of gone. And remember that doctor said he was able to cure most of them. And he doesn't know why, but every person is different. That one case didn't work out. Masticophora means flagellants. That means all these organisms have flagella. First one, trypanosoma. Looks like a ribbon, but he's between the red blood cells. He's not eating them. He's in between them. And I'm going to scroll through some pictures from the time comes. Two diseases, Chagas disease and African sleeping sickness. Geographically located, certain vectors. No one's associated. Chagas is where? As a kissing bug, it's located where? Central South America is also in Texas and California. African sleeping sickness, Africa. But what is the vector? Kissing bug. Okay. Ribbon, and this ribbon can twist. I can take a ribbon, twist it, and what does it look like? Red blood cells. Triple moment. Sort of like a pear shape with some flagella, sexually transmitted. Find him in the earwig. Females, focus PSC, males, asymptomatic. Okay, Giardia at this point in time is a prevalent disease, diarrhea in the United States, fresh water. Remember the old man? The two nuclei are in the eyes. You have a bar for the mouth. Sometimes you can see the flagella to make it. Sometimes you can't. You say you want to know what diarrhea, prevalent in the U.S., in water, and in the Mexican area. And I'll think because you're in a remote area, you don't have a parasitic disease. Okay, apopcomplexus has the apex structure. The worst of all. What is that disease? Malaria. That's a reoccurring cycle. And in fact, each substratum of malaria, plasmodium, has a certain fever cycle, 4 hours, maybe 8 hours, and then stays still. These live where in the large group? Using red blood cells like a guide helps give you a function of the right organism. Inside the red blood cells is the plasmodium that your ring, basket, can completely fill it, okay, or your bar. Look inside the red blood cells. Outside red blood cells, switch over to maybe things like medicine. Okay? And it's been eaten. It could be, and it's not eaten. Think about red blood cells in the diaphragm. Okay, cycles of fever, malaria, carried by what? The anomaly of the fever. We'll say anomaly. Toxoplasma, have cysts, have little boats that break out. Remember, parasitic protozoa have a circular life system. There's a cyst which is dormant, and what's the name of the feeding form? What's the name of the feeding form? Feeding form, protozoa. So like cats, you saw the thing about poor rats that don't donate themselves to cats. Dangerous prey for them because it can harm blind and or harm humans. Critical meridian, one of the ask you to know right now, dramatically associated with AIDS. That gets these. Moving over to our one protozoan, cilia is the lanthium. Technically the lanthium colon. Big giant ball. You might see a little halo around them, which are really the cilia. And it will have a bar. It doesn't disease. And the bar is twisted. Can I give you an example? Think about a horse. You show somebody a 10-toe horse. Say, that's a horse. Well, they go out and they see a Shetland pony. Or they go out and see a palomino. Or they go out and see something else. That's not a horse. No, this is a form with all the other variations as well. A little bit close in. Okay? Those are your parasitic protozoans. Parasitic protozoans. Going back to my typical test, we'll give you a break before we head into the worms. I watch my time, several times as I do this. You remember what the MTIC test is? Relation
citrate. And I'm sure why you're waiting for this one to make a surprise, but it's positive endomethylene. Negative, most toxic citrate, no, not the ion, just make it word. That is the pattern for the salt. And they have databases. We have this, no one wants to move over to work.
So we have to know what that stands for.
Yes, it's what it stands for. Endomethylene,
Now, heading for the worms. Three groups. You have trematose, which are your flukes. Nematodes are your mealworms. And up here are your cestodes that are your tapeworms. Flukes are flat. Think of a leaf. Flat as a leaf. Able to squish in between tissues very well. Two examples we have are schistosoma, okay, and fasciola. Schistosoma, its egg has that spike. It has intermediate life cycle in the water where it drills into the scale, lives for a while, comes out as a swimming sicaria. That sicaria can do what if you're walking through the water? Drill straight through your foot, drill through your skin. This is often a problem where Asia, rice paddies, of course, it can hit anybody in there. Then, of course, it's going to head and affect chickens more. Fasciola, technically Fasciola hepatica, it has the egg that looks like an egg when you tapped off the top or they had a cat. It is the liver fluid, of course, but it's fat. Hepatica, the liver. This is the one, remember the film where they showed the meat instead of the bladder worms living on it? Don't eat it. That kind of stuff. Now,
Is that actually where the worm comes out of? Like, is that the cap that
the worm
just comes out of the top?
Yeah, this usually just floats away. That breaks off.
Fair enough.
It can also be a larva. Okay, that is your trematose. You've got more nematodes to know. The trematose. Enterobiotis, Tricurus, Decatur, Ascarius, Trichonella, Filaria. No scientific name, common name, because we use English when we go to another area, to say the scientific name, you're going to know it. Enterobiotis is your pinworm. Its eggs is an oval, that's sort of flat on one side. Sort of flat. You would look on the testing card with keywords such as intestinal infection, comes out from the anus at night, lays eggs, itches like mad, often children will self-infect. You can test it with Scotch tape. What's the Scotch tape for? Take off the eggs. Probably a good one. First, just let me notice the lemon, the bulbs morphia, that's your whipworm, it's much smaller. Decatur is your hookworm. The eggs are not as definitive for identification, but that's the one, if you go barefoot in the south, it can get into your foot quite easily. Ascaris doesn't have a common name at this point in time. Probably life cycle is in your lung, and it can get pretty big. There are some samples back there in jars. It's a scalloped type of outer shell to the eggs. Sometimes hard to see because they break apart. Trichonella. In what? Pork. Okay? Where will you find it in a person? Or in an egg? Slice what tissue? Muscle tissue. And you're going to find the world either cut this way or it's on its side, coming out at you, in which case you will see holes coming out at you. Filaria gives the dog heartworm. So with the heart, we have a jar back there, carried by what? Mosquitoes. We get elephantiasis. It blocks our lymph system, and therefore what? It causes massive, no drainage, you get massive swelling. Very good. Okay, last one, gotta get out of the way, is the cestode called tania. That's your tapeworm. Now the scolex is the head. It can have sucker structures on the side, can't have hooks. The segments coming off the head are called proglottis. The proglottis have male and female sexual organs in them, and they produce bags of what? Eggs. Don't forget that. Now remember this. That's tania. What was tania? deer tick, Borrelia, Lyme disease. And what is not a vector, but actually will eat through your skin, called massive itching. Scabies. So know your vectors. And you're going to have to change your powers if you look at them today so you can see them. You don't want to see a little spot inside, you want to see the whole rose. Do you have any questions about any of this? Okay, I'm going to stop right now, which I'll ask you questions later. I'm going to do lab cleanup.